Hi folks, today I want to talk to you about the process of engineering fungus and then hopefully do a series of videos through all the steps up to the point where I'm at and get you into real time so that you can follow my progress and then you can use that to help you do your own research. So let's start with looking at some fungus. So I have some growing on a plate here. This is some aspergillus that actually grew in my local environment with a colony of penicillium here at the top. You might ask, how do I know that these are aspergillus and penicillium? Well, one, when you look at molds long enough, you begin to see certain phenotypes or characteristics of the organism that give it away. The telltale green color and the aspergillus, if you could see it up closely, you'll see little dots. Those are going to be the conidiophores, which have a very characteristic look, especially under a microscope. In fact, this will probably be the first organism that I do a genetic transformation on. And that's not really new or novel. There have been a lot of people that have done genetic engineering of aspergillus, in part because aspergillus produces some pretty interesting secondary metabolites, certain species of it anyways. And so if you can get genes into it, you can ferment them at large scale and pretty cheaply get them to produce some really valuable, interesting things. As an example is there's a species of mold called trichoderma, and scientists have figured out how to put the genes in it to produce a milk protein. And as a result, they're able to ferment a lot of this trichoderma and have it produce this milk protein and use that to produce dairy-free alternative foods. Pretty cool stuff. So when we look at this mold, this aspergillus, we know just from science that everything living has DNA. We know that every cell of this aspergillus species has DNA in it. And so the question is, how can I modify that DNA? Where do I even begin modifying that DNA? What I'd like to do on this video is just give you a brief overview of some DNA stuff and talk a little bit about DNA plasmids, because that's probably the most difficult part of the entire genetic engineering process, is designing the vector that you want to go into the organism. All the lab skills and things like that, what I've learned is you can learn those pretty easily just by practicing, and you can do them in a low-cost environment. But the hard part, as usual, it's sort of like computer science. It's sort of like if you want to write code, learning to write code is easy. A monkey can type keys in a keyboard, but knowing what to type is the hard part. So with genetic engineering, the molecular tools you're going to use to get the DNA into the organism, not that difficult, but the hard part is designing the code that's going to go into them. So we'll talk a little bit about some of those, and yeah, we'll go from there. I have in my hand here an artist's rendition of the DNA molecule. Usually when you see DNA, you'll see it as a sort of double-stranded thing, and you might see these look like rungs of a ladder that kind of connect between them. And really the difference between all of these is the amount of information being shown. It's a long molecule. It has two strands, and then it has, these connections between them. Here's a more detailed view of what the DNA molecule looks like. We talk about those letters, A, G, T, and C. You can see here that actually translates into the individual nucleotides here, and that's where they get the names. You'll see here that if you were thinking about that ladder analogy again for DNA, that these would be the support structures on the sides. These are the phosphodiester backbones, and then we have the individual nucleotides connecting them in the middle. So in the case of our aspergillus here, somewhere in each one of the cells is a nucleus, and inside of that nucleus is the chromosomal DNA for that organism. So we have to figure out a way how we can modify its DNA so that we can get it to do what it is that we want it to do. Although a lot of the information that we're talking about here applies to just fungi, the principles here of genetic engineering apply to pretty much every kind of cell, whether it's human or it's fungus or it's plants or animals. These are all DNA-based life forms. And when we talk about genetic engineering, we're talking about how to alter that DNA using molecular biology tools. Now, in this particular tube, we have some E. coli growing in LB media. And if you'll take a look at this, you'll notice it's quite cloudy. And that's from all of the cells, all of the bacteria that's growing in solution here. And this is the DH5 alpha strain of E. coli, which is pretty commonly used in what's called molecular cloning, which we get to later. But what's interesting about these bacterial cells is, well, bacteria are called prokaryotes, so that means they don't have a nucleus like the fungal cells do. But even more interesting about E. coli and bacteria in general is they have a extrachromosomal area of DNA called plasmid. So think about that for a second. Bacteria, just like any other organism, in order to continue functioning and to replicate, it has its own machinery for processing its DNA, for replicating itself, and for producing another cell from its own chromosomal DNA. But it turns out that bacteria also has DNA that replicates independently. This extrachromosomal DNA is called plasmid DNA. So imagine a bacterial cell, and in one area you have the chromosomal DNA, and this is the part that gets replicated and used to produce copies of the cell and for the cell to function. But in another area of the cell, there's a circular piece of DNA in there that replicates independently from the chromosomal DNA. It usually doesn't have many genes in it. It usually has a handful of genes. So why is this important? Well, at the heart of a lot of genetic engineering is we're going to be tapping into E. coli's ability to replicate these plasmids independently in order to do a lot of really cool things with genetic engineering. To illustrate this idea of plasmids, what I have in my hand is a typical extension cord, and I'm going to connect it here so that this cord is connected into like a big circle. You might go, well, it's not really a perfect circle. Well, keep that in mind, because there are something called Plasmid Maps, which are just these illustrated guides to tell you what a plasmid does and lays it out, and we'll come back to that. Often plasmids are presented as these perfect circular things. In reality, they probably look a lot like this, and they're probably coiled. There are certain key elements of a plasmid that are necessary for plasmids to replicate. One of them is called an origin of replication. You might see that if you look at a plasmid map. It's an area of DNA that kind of tells the cellular machinery how to keep replicating that plasmid. If we took this cord and we color coded it so that we labeled the individual sections here, let's say this is a gene, this is the origin of replication, and there's a bunch of letters here that represents the origin of replication. We might have another section here with our gene of interest that we want to insert or make a copy of. We might have another component here. It's not really feasible to represent what this circular piece of DNA looks like using an extension cord. And so what we can use is something called a plasmid map. And the plasmid map gives us a nice graphical visual way of understanding what's in a plasmid, what genes are present, where they're physically located. So let's take a look at a plasmid map that's in a paper that I've been using that is used to modify fungus. And notice the circular. We had the extension cord before, but now here's a proper circle here representing the plasmid. Now there's some things about this particular plasmid map that are missing. This doesn't show origin of replication or the essential machinery necessary for this plasmid to replicate inside of E. coli, but it kind of gives you an idea of how plasmids are represented in the literature. And a plasmid map is going to denote a couple of things. It's going to note genes in here. So each one of these, this is a GFP gene, this is a hygromycin resistance gene and a kanamycin resistance gene. And it's giving us some other parts here as well. And what this really translates into is about just under 9,500 base pairs of DNA. And this gives us a map of knowing, okay, if we start at the very beginning of those sequences, the 9,000 base pairs, what sequences belong to what functionality within the plasmid map itself. There are other ways of representing what's in a plasmid. For example, here's a plasmid that I'm designing for engineering mushrooms, and this is actually only showing a linear section of that. Imagine that this little section here will actually be contained in this particular area of this plasmid over here. So let's look at this Picambia 1300 plasmid. First of all, plasmids, you can find them online and you can get them from add gene. And so the convention for naming these plasmids is they'll always start with a P that denotes the plasmid, and then they'll have another name for them after the fact. But PKMia is interesting because we're going to be putting this into two different kinds of bacteria. And so it needs to have the machinery to support both species of bacteria that we're going to be putting it in. We're going to be putting it into E. coli, but we're also going to be putting it into Agrobacterium tumefaciens, two different species. So they need a little bit of a difference between them in order for this plasmid DNA to replicate independently and autonomously within its own cellular machinery. So this is called a binary plasmid. And so if we look here, we'll see that there's this left border, and we'll see over here that there's a right border. This kind of denotes the area of DNA that actually will get transferred to the host organism. In our case, we're going to be modifying fungus, and we're going to be putting our genes inside of these border regions, because when this gets into the agrobacteria and we put the agrobacteria next to our fungal cells, agrobacteria has a mechanism for transferring this area of DNA to that host organism. And so by far the most difficult part of doing genetic engineering is designing the DNA, because to make changes to an organism's genome, there are a lot of critical components that you need to know before you make modifications to it. Identifying the genes that perform certain functions within the organism, that's an ongoing area of research. And as sequencing goes down and we are able to collect more genomic data, we can use machine learning and we can use large-scale comparisons of organisms to try to hone in on new genes that produce amazing new compounds, for example. But in addition to the genes, there's also machinery before and after the gene that's necessary to get the organism to produce or to actually process the gene that you're interested in. You also need something before the gene that tells the cellular machinery to start transcribing this gene and to produce it. In some cases, you might want it to produce a lot of it. That particular area might be referred to as transcription factors or a promoter region. And it's an area of DNA that goes before the gene that tells the cellular machinery that, hey, there's a gene here, you need to start transcribing it and producing the protein. At the end of the gene is a terminator region, which basically is kind of the opposite of the promoter region, and it tells the cellular machinery to stop. And so if you're going to engineer an organism which hasn't been engineered before, there are a few things that are challenging, but that you need to do in order to be successful. One is you need to identify some common promoters that will allow you to insert genes and ensure that the machinery for that organism can properly transcribe the gene that you're looking for, and terminators. And what I'm actually seeing in biotech is a trend towards automating the lab side of it, having people that are really good at the genomics and data side of it figure out the genes and figure out the experiments to go into their plasmids, and then they can write some code, send it out to a third-party provider that will run the experiment in the cloud, get the data back from an API. We're almost to the point where you can start a genetic engineering company and you don't really even need a lab. There's going to be companies in the future that exist now but are probably going to continue to grow, which are completely robotic and automated driven, that will run these experimentations for you. And all you're going to be doing is focusing on genomic data. And I think that's pretty cool. Although a lot of people think the molecular biology side of this is the fun part. It's the part that they kind of associate with genetic engineering. What's probably going to happen is you're going to have more and more people specialize in designing the constructs, the things that actually go into the organism, and then outsourcing the molecular biology work, which is cool. It's going to allow things to scale and move a lot faster. So that's a brief overview of DNA plasmids and how they fit into the broader picture. It's really the payload that's going to be going into the organism that you want to modify. And it's a really important and big part of the genetic engineering process. So whenever you see those little round circles, now you know that that represents a plasmid, and you know that all of the little things coming off of it are just annotations telling you really important parts of the plasmid. And so in my case, I have a plasmid because it's known to already work. I'm using that plasmid to do my first fungal transformation. And then on the whiteboard behind me is a plasmid I've already started developing myself for future mushroom engineering experiments. And that's, as I said before, that's where everything begins. It starts with designing the DNA construct. And arguably this is the hardest part because everything downstream depends on you designing the DNA correctly. And if you make any mistakes and your experiment doesn't work, it could go all the way back to the design of the DNA that you put into the organism. Probably in the next few videos, I'm going to show you how we can take a little bit of plasmid, we can put it into the E. coli, we can grow a bunch of it and filter out the plasmid from that so that we have all that we might ever need. And then we'll continue on the process of genetic engineering a fungus. So if you have any questions, let me know. I'll try to get to them in the comments. And if I made a mistake, please tell me. I'm always looking for feedback. And I've learned all of this in the last year, so it's going to be imperfect, but bear with me. Okay, thank you guys.